to everybody. It's a pleasure for me to be here. I would like to talk about what we do at Limmatech and our goal and vision. So in a snapshot, what is Limmatech? We are a life-driven company that we are really bringing novel idea into clinical proof of concept. We are very good in translating these in a short and good risk-taking manner. And we are now in a phase of a spin-out of the novel platform into a new company from the Limmatech, that is a mother that is well-financed with big collaboration. And now we are here to look for long-standing investors in order to bring us to the next phase. So who is behind the Limmatech? It's a management team that has worked together for many years in creating new innovation platform. And we are working together since many, many years, and we are surrounded by a worldwide expert that we use depending of our needs, from the clinical to the development as well as from the scientific part. So we really have a track record of many years of bringing innovation into clinical proof of concept through all phases. We really like to take a smart risk, and we have been working with collaboration and non-diluting funding until now. Today I'm going to talk about a new platform that we would like to spin out, and all started with the problem, and the problem is about the CHO cells. We know that CHO cells is very good and are used to produce glycoprotein, but there is a shortcoming, the fact that it's very difficult to control the glycan. And more is known that the glycan that is decorating your protein is very important for the functionality of the protein. Could be from the potency, from the immunogenicity, but as well from the stability. And this has been known and now published more and more. We believe that with our novel platform, we bring the solution. So we are having a new cell line that is, first of all, much more simple. So this should bring as well a better cost of goods, something that is as well very expensive, for example, when you think about monoclonal antibodies. And in addition, you can really control and make a homogeneous glycan that is able to enhance the efficacy of your protein. A little bit in more details, in the next slide, is really two main features. One from the manufacturing is because we start already with a very homogeneous glycan that we build up. In addition is the site occupancy, and this brings big advantages from a manufacturing point of view. From the effectiveness, we are going to targeting two different modes of action. One is because our glycan don't have the focus, therefore we are going to enhance the potency of our glycoprotein. And the second mode of action is because we are going to build a human glycan with the sialic acid that is known that will prevent immunogenicity of your glycoprotein. So these two big advantages differentiate to all the other platforms that now are out there. And this as well opens up a lot of opportunity with different products, because depending of your glycoprotein, what do you want to target, you can make your homogeneous sugar in order to enhance the property that you would like to have. And then can be applied in different indications, such as enzyme replacement or in monoclonal antibody for cancer, inflammatory, but as well to more complex glycan until blood protein. Where do we stand now? We are focusing on monoclonal antibody and for the inflammation. So because it's a platform that can be applied, we have a very careful IP strategy. On one end, we want to have a very broad IP for the technology, but as well then try to have a very specific IP for the product itself. And already now, since the conception we started, we have filed already 3 patents and 2 are ongoing. This brings me to our leading candidate. We choose an anti-TNF-alpha for the inflammation, and for 2 main reasons. First of all, because for anti-TNF-alpha still there is a big market. And even if there is a lot of competitors and new biosimilar coming up, this increases the opportunity of this molecule. In addition, is because we know that if you come with a molecule that differentiates itself and have a higher effectiveness, you can really make a difference and take a share of this market. Second, we want to go with a molecule that is already known, because we want to demonstrate that the platform, with a head-to-head comparison with a molecule that is known, we can really make a difference. And when we show this in a phase 1-2 study, then this can be applied to all the other molecules of, as I showed before, that you can use it with this platform. So how do we do the first leading candidate anti-F-alpha? We go to IBD, inflammatory bowel disease. Why we go there? Because it's the perfect way to show what our platform can do. On one hand, in IBD, there is still a big unmet medical need. The anti-TNF-alpha, that is the gold standard, still is only 60% efficacious as a primary treatment. In addition, with the long term of usage, because it's a immunogenic protein and produce anti-drugs antibody, you're going to lose the effectiveness of this product. And then the people, either they have to increase the dose, either they need to change to another molecule. We believe that we can target both with our modification of the glycan, homogeneous, because without the focus, we are going to increase the effectiveness and the potency of the molecule, so the primary treatment. And in addition, because we have the sialic acid, we are going to increase the long-term continued response of this molecule. What is the patient benefit? Is that we can have more people that can be affected by our molecule. In the long term as well, lower dose, less injection. These are some data that now I can show you. There is more. But for example, this is an in vitro study where you can show that we compare to the Humira, the gold standard, that through the alpha-cosylation for this homogeneous sugar, we can really enhance the functionality of the potency of your molecule. And we show it in two different ways, with the NK cells, but as well with PCM cells. And you can see that we have a tenfold increase of the potency of the molecule compared to the Humira, the gold standard. So where are we and what we want to achieve? So right now we are making the selection of the leading candidate that to bring into the clinical phase. We are going by the end of the year to complete the preclinical package, to make a pre-IND as well, because this is a new cell line and we want to have the feedback of the FDA that this will lead us to the GMP production and enter into the clinical phase in 2022. The beauty is that because anti-TNF-alpha is already known and very well used, is that already in a phase 1-2 we can to have a head-to-head comparison in the population, in the IBD population, and obtain efficacy data that will be available in 2023. So in 3 years of time you can really highly de-risk your product, but as well the platform itself. So some financial assumption, we are going to, from the mother Limmatech, we are going to make a spin-out of 14 people. These people will develop this platform further and going to bring this first molecule into proof of concept. The model is with outsourcing preclinical manufacturing and the clinical trial. We are going to use the mother Limmatech for additional resources. And as I mentioned before, in 3 years, we expect to have efficacy data. We are looking to do all that. We are looking around 25 million. We are now starting our Series A. And these 25 million will bring us not only to the proof of concept efficacy data of the first molecule, but as well to further develop the platform from a manufacturing point of view, but as well to fill up the pipeline with other candidates that could be applied in different sectors. So About an exit, we think I don't like to talk about exit, but more value creation. We believe that this will bring a big jump to the next phase in these 3 years, where we can continue to bring this product to the market if we differentiate to the competitors. And as I say, there is still market there, but as well then to start to develop the others. And so we have, after this value, you could have seen as partnership, license out, as well as acquisition. So to summarize what I just said, I believe that we are an experienced team, very known in glycoengineering, but surrounded by worldwide experts to support us in the things that we need to do. We are entering with our leading candidate in a market that is very big, and there is still space for a differentiated molecule. We are having an early clinical proof of concept that can really de-risk your platform, and this with efficacy data in 2023. And now we are starting our fundraising, Series A, and we are looking for bringing this molecule to the next stage, and the company as well. Thank you very much for your attention, and I'm here for any questions you may have.